Structural characteristics of the brain and their relationship to the behaviors of corvid birds, parrots, and sharks by Jori, Tytus, Barron, and Avina.
Species exhibit different neuronal density, which is a result of localization over millions of years. These findings support the theory that development and size of a region of the brain could indicate different advantages and functional uses. Our project will be conducting a lit review of papers in order to compare and explore the localization of function in the brain and how their sizes and functions differ from species to species.
First, we'll talk about the corvid bird, or the crow. So how are they so intelligent with only a 10-gram brain? Well, their cortex, or pallium, is far greater than that of other birds. And because of their very small neuron size, they have a potentially high neuronal density. Also, their structure of the pallium is in a nuclear arrangement. And why and how do they have such complex cognitive ability? Well, they are very adaptable to their environment and face and overcome different challenges on a day-to-day basis. They exhibit causal reasoning with tool manipulation, mental flexibility with their ability to abstract rules and relationships between things, and imagination through the use of a cognitive map.
Next, we'll move on to the parrot brain. How does neuronal density influence the parrot brain? Compared to the mammalian brain, the parrot brain is smaller in size and has less cortical folding. However, it has more cognitive power per pound due to its increased neuronal density. This allows for more connections and stronger responses. As a result, the parrot has better use of tool manipulation, increased flexibility, as well as increased alertness.
Now we will talk about the shark. Evolving from the brain of a lamprey, shark brains developed to include the localized areas known as the cerebrum and the olfactory bulb. Although the shark brain branched off early in evolution compared to the human brain, many structures of the midbrain and hindbrain can still be identified in both. By electrically stimulating areas of the shark brain, researchers have identified that different areas correspond to different physical responses from the shark's body. Additionally, by continuing to study the localization of functions through stimulation, researchers can begin to propose specific correlations between the structure of the brain, the behavior of the animal, and a species' potential for cognitive ability.
In conclusion, in this paper we show that brains have similar regions and function localization across species. And because of this, we are able to isolate and study areas of the brain across species to gain a better understanding of what parts of the brain lend themselves to higher intelligence. We find that the structure and neuronal density play a big part in the determination of higher intelligence. However, apart from that, complex cognition evolved from external factors also plays a big role in the animal's intelligence. Thank you.